Good morning, everybody. I'm Myra Ansari here for Wave Now. We are going live to UofL Health. They started a press conference just a few minutes ago, but they're announcing nearly $12 million in NIH funding for research studying links between microorganisms and disease. So folks are getting together right now. Let's listen in.
Thank you, everyone. As Dr. mentioned, I am a graduate of the COBRA program through the first cycle, and I started the university here at the University of Louisville in 2016. And as a junior investigator, the COBRA program was actually instrumental in helping me set up my lab by providing both direct support in terms of research funding, but also, more importantly, a formal and informal mentorship structure that I could use to leverage my career further. In addition to the core facility run by the COBRA program, we have access to cutting-edge technology and innovative approaches to augment our research. I'm a molecular virologist by training, which means that I study the viral interactions between the host and the pathogen through protein-protein interactions or DNA-RNA protein interactions. And this interface is critically important because this is where we can discover new drugs for antiviral therapies. But what's also important is keeping this interface in the context of inflammation and disease, because really that's what we care about, is identifying the next generation of scientific discoveries that are going to allow us to leverage that interface to better human health. In addition to the research training that I received and the mentorship that I received, we're able to trickle that down to our actual trainees within the lab to help support them in their status as learning scientists. And so the COBRA program has been instrumental in my development, and I can see that it's also instrumental in the development of those at my peer level, above and below. And without the COBRA program, I can certainly say that my lab would not have reached its own independent funding through an R01 grant from the National Institute of Allergy and Infectious Disease. With that, I'd like to welcome Dr. Badalori.
Thank you all. So this program got started actually with a university support of over $750,000 given over a 3-year period to initiate microbiome research. So the main thing that I would like to emphasize here is, unlike resource-rich universities like mostly in the Northeast, state of Kentucky, is considered that we do not have as much of research funding. So this particular program, the Centers for of Biomedical Research Excellence, provides a substantial bolus of funding to train junior faculty with the support of senior mentors, as well as, a wide range of colleagues, and bring diverse fields together. So in this context, when we came together, the Oral Immunology and Infectious Diseases Department and Microbiology and Immunology, as the name suggests, they're very close, do very similar things, but then are completely separated both physically or administratively from dental school and medical school. And this program, the university's program, first got us together and allowed us to bring in, do some basic work and bring people together to apply for this about 6 years ago. And as Dr. Lamont already spoke about the tremendous success of the program, which over the last 5 years produced 6 graduates, meaning 6 different individuals getting their R01 grants, and we currently have 3. But all of this, again, goes back to one central theme compared to resource-rich universities. This program also provides a substantial sum of money for shared resource development, which is one of the called core facilities that I run for the department and also for this particular COBRA. These core facilities bring state-of-the-art technologies like, intravital microscopy, microbiome analysis methodology, new sequencing technologies, as well as what we just got 2 supplements for the COBRA. In addition to the $11.7 million that we already was awarded 6 months ago, in the last 2 months, we got $900,000 additional dollars. These will support something called chip cytometry, which is a spatial biology where you could look at the single cells on slide. And we also got a project for team science. This is between Dr. Lorenz and myself to look at how inflammation and immune responses are integrated in both long-term and short-term fighting infections, as well as many of you may have heard recently in the last week or two, every day there was a story on crystalline silica exposure, in the news, from West Virginia coal mines to California, where they prepare kitchen countertops. About 40 people died in California, young people in their 30s, because they exposed to this crystalline silica. And this new supplement would allow us to work how the mechanisms and the details of the disease development. And so in this context, again, I would like to emphasize the importance of team science, because between the 2 departments as well as the number of people. And the idea that the NIH is willing to support this kind of a large projects, which usually are reserved for major universities, and so that the ones that are not at the highest levels can also come up. And this has all been possible because of excellent support from our Executive Vice President of Research, Dr. Gardner's office, because they provide us, in addition to these NIH monies, more than $1 million over a 5-year period. That really strengthens the program. And with that, I would like to ask Dr. Gardner, who is our Executive Vice President for Research, to come and say a few words, especially as it relates to retaining the faculty here who are all new graduates. Once they get their R01s, how do we keep them here and sustain this research enterprise? Thank you.
Thank you, Dr. Bodolori. I'll give you your cash later that we agreed on for you to say nice things. Congratulations to you, Dr. Lamont, and to the entire team. People that work on these know that there's a lot of people besides those sitting up here that are instrumental in actually accomplishing this and getting these types of grants. This kind of grant is transformational in enabling our ability to do groundbreaking research that can have really broad impact, that can understand diseases in new ways that lead to new types of treatments and can improve human and animal health. How our microbiome influences our health is incredibly rich and complex field, and new discoveries by this team and others are uncovering these relationships all the time. And I think, as Dr. Bodolori said, we've all heard about it in the news, about all these new ways of understanding Alzheimer's disease and cardiovascular disease and other types of things like that. The University of Louisville is characterized as a research-intensive university. And that means that the university has a mission to discover new knowledge. In addition to transmitting knowledge to students and trainees and so forth, it's new knowledge that's one of the major missions of this university. And we have a commitment to creating new knowledge, and that's what this type of grant enables: the exploration, innovation, finding new solutions to important problems that have impact far beyond this campus. And furthermore, our office and our university are committed to bringing those new discoveries and new types of treatments to the world, to the clinic, to the bedside, to improve human health in the end. This major grant led by Drs. Lamont and Bodolori is a great example of the university's commitment to new knowledge discovery and bettering the human condition. Congratulations to you both. Congratulations to the entire team that it took to put this together and to do this amazing work. Thank you all again for being here and celebrating this achievement. And I'll turn it back over to John Carmen to field any questions. Thank you.
Thank you, Kevin. Yeah, that concludes the formal part of our program. If there's any questions we can take from the podium now, we can do that. We can also make people available for one-on-one interviews after we wrap up. Anything?
They mentioned research. What are some of the things this research has already discovered that you're excited to continue working on with this grant money?
Please come to the mic.
One of the things that we are interested in is discovering new host pathogen interactions. So viruses are made up of nucleic acids, which encode the information to essentially make more virus. But parts that are used to assemble the virus are actually delivered in conjunction with those genomes or those nucleic acids to the host cell. One of the things that we're actively working on is the contribution of a part of the viral particle that's actually delivered to the host cell that then comes apart. That's a long time been thought to not really have a function afterwards. But we've actually determined is that it interacts with a very important signaling protein within the host that's key to the regulation of the innate immune response. So essentially, in addition to delivering the genetic information to make more virus, this protein-protein interaction, this novel protein-protein interaction discovered through the COBRA program, is actually causing the host's innate immune response to be dampened or weakened until viral infection can continue. This is an excellent example of a potential target for a drug therapeutic, because if we mutate this interaction or prevent this interaction from occurring, we no longer see disease occur. And so if we can develop a small drug or a small molecule that targets this interaction, that's a way that we can actually take science from the laboratory bench all the way to the bedside to potentially impact public health.
So break that down for what like an everyday person to understand.
Yeah.
Like, how would somebody with no science background, how would you explain that?
So you can think of interactions between the virus and the host as a lock and key.
Is the host a human?
The host can be a human. In this case, it is a human. It's typically SARS coronavirus, can be any of the alpha viruses. We work on alpha virus models. Viruses that cause polyarthritis or encephalitis in young children and the elderly. These are North American diseases with a mortality rate of around 40%, depending on which member of the family you're looking at. But you can think of the interactions between the host and the pathogen as a lock and a key. And really, in order to open that lock and cause infection, that key has to fit snugly and it has to fit perfectly in order for it to turn. This interaction is essentially an example of that lock and key, where we may actually be able to put something into the lock to prevent that key from being able to enter and turn. And so that's a way that we can essentially prevent a critical process for the virus to engage with the host to enable infection and the formation of disease after somebody's appeared in the clinic.
That might be an oral drug that you take and have in your system.
Oral
If you get
intravenous.
exposed to a virus like SARS-CoV-2,
Mhm.
it would not replicate in your body.
Yeah, and one of the nice things about this, or one of the importance of antiviral drugs or antiviral targets, is you have to ensure that they work after somebody goes to the clinic. Right? People don't go to the clinic when they're healthy, they go when they're sick. And for the diseases that we work on, for the alpha viruses, when you go to the clinic, it can often be too late, particularly if it's an encephalitic alpha viral infection. Usually, if you end up with clinical encephalitis, there's about a 40% chance that you will not leave the hospital. So if we can have a therapeutic that actually works at that late stage by targeting the innate immune response and the inflammatory process involved in the formation of disease, that's a way that we can potentially shift that needle towards recovery versus the dire consequences of infection.
I could add a couple of things, to that. One is historically, inflammation is considered, or last 20, 30 years, is always linked to all the diseases or inflammatory diseases, so whether it is heart disease, Alzheimer's, and everything. But the underlying principle is that inflammation is good when it helps remove the underlying cause, whether it is an infectious agent or whichever. So some of the work that we have been doing over the last 25, 30 years really points that very clearly. So early on, if you have an inflammatory response, it will clear the thing and then you restore normalcy. But if you change microbiome, change your diet, or these kind of things leads to alterations in your microbiome, which means the inflammation persists. And the underlying cause never goes away. And that is one of the things that we were able to kind of get latched on with this COBRA, with, again, a supplement about 4 years ago for doing Alzheimer's research. I mean, so to develop new models for Alzheimer's disease. And we found some surprising things in those studies. For example, a receptor that normally kind of initiates inflammation. When it is removed in a mouse model, what we found was the Alzheimer's disease is highly accelerated. So the reason for that is the normal inflammatory response is critical for removing the amyloid plaques. And if you don't have that, and the insult continues, the amyloid keeps forming, you develop a degenerative neuro disorders like Alzheimer's, Parkinson's, and many others. So what comes out of this study is you will have to think about it radically differently when approaching these things. And there may be, especially for Alzheimer's, there were no drugs. I mean, there is only one that has been approved in the last year and a half with no significant clinical benefit associated with it. But at the same time, there may be literally hundreds of drugs for other inflammatory diseases that could be repurposed if we understand the basic mechanisms of the disease, as well as how we should kind of target those. So we work on one of those kind of pathways called the leukotriene pathway. I mean, so these are lipid mediators of inflammation. And this applies not only for those type of inflammatory diseases, but for some infectious diseases as well. I mean, that's what Dr. Lorenz's lab works on. Yersinia pestis, the causative agent of plague. I mean, and this bug has learned to suppress immune response, I mean, through certain pathways, activating those. Thereby, the initial response is not there. By the time the immune system recognizes it, it is too late. That's one of the reasons why we have all of these new funding avenues to go and target how to tweak these systems or improve our understanding of these pathways.
All right, any other questions? Okay, we will have an opportunity for one-on-ones if anyone is interested. Now I know we've got a quick photo here in the front, and there'll be the opportunity to tour the labs. Thanks everybody for being here today. Please be safe and try to stay cool today. Thanks a lot.
Long time no see.